So, if we look at homeostasis, homeostasis normally comes from two words, and the two words that have been combined to form homeostasis, that is home and then stasis. The word stasis comes from the word static. Static, it means that we are keeping something without changing it. It is maintained at a certain level. We are keeping home static. So combining two words forming homeostasis. So homeostasis generally looks at maintaining the home static, or the word static can also mean constant. So maintaining the home constant. What is the home? The home means our bodies. The home means the bodies of other organisms. For example, maybe cows, maybe chicken, whatever. Any living organism, its home to be maintained constant. That is what we call homeostasis. So that home is going to be the internal environment. So the maintenance of the internal environment constant, that is the meaning of the word homeostasis. Coming from two words, that is home, that is, for example, your body, inside your body, and then stasis, maintaining at a specific level. Why at a specific level? It is at a specific level because that is the level which is desired by the body for your body to function very well. There is a desired level to which each and every kind of need must be maintained at up to a certain limit. For example, you cannot you cannot drink a full jerrycan of water at a single time. It is impossible. There is always a level that you are going to stop on and you feel like you are now satisfied. There is a certain level of food you are going to eat and you feel you cannot take any more. So that level of that you can always stop on, it is the level through which even the body must maintain whatever is required by the body at a certain level, through which the body must continue so that it can be able to process its activities, it can maintain functioning very well. So which factors must be kept constant? Of course, when I talk about factors that must be kept constant, we look at what must be maintained, not changing in the body. Of course, we may not have a certain value which is specific, that this is the value that must be maintained, but it can be at least a range. It can be a range. That if it is a range, for example, if I'm consuming 1 kilogram a day of posho, but there must be a range that maybe I may consume a half to a kilogram a day. So, but below a half and beyond a kilogram, I may not manage. So that range that must be maintained constant, it enables the body to continue functioning very well without finding any challenges. So let us begin with factor number one. Factor number one, that is the blood glucose level. Blood glucose level, whatever food we are taking in and it is in the form of carbohydrates, it can increase the concentration of glucose. So it implies that if the concentration of glucose is increased, it must not go beyond what is required. What is required by the body, it is around 90 milligrams per 9 cubic centimeters of water. So if it goes beyond that, it is going to cause abnormalities. Remember, we have three types of solutions depending on the organism that is or on the body which is within that solution. That is the hypotonic solution. We have the hypertonic solution and the isotonic solution. So the hypertonic solution has a higher concentration of solute than what is surrounding it. So if the concentration of glucose goes high, so the solution which is going to be surrounding the body cells is going to be hypertonic, implying that there is a possibility of those cells shrinking due to the loss of water to the surrounding cell, to the surrounding environment, or to that solution. Then there's what called isotonic. Isotonic, the concentration of the solution is equal to the concentration of what is within the body cell, the cells. And then hypotonic, the concentration. So in isotonic, it means that there's going to be a net exchange. Net exchange, it means that we are going to be having the same water leaving, the same water entering. So the cell can remain normal at that stage. Then we have hypotonic. Hypotonic, that is when the concentration of the solids which is in the cells is higher than the concentration of the solids which is in the surrounding environment. For example, in the solution, that is the surrounding environment. It implies that there is a possibility of too much water running into the cell by osmosis, implying that there is a possibility of the cell bursting. So it implies that we must maintain the concentration of glucose at an isotonic level, whereby there is always a net exchange of water in and outside the cell. Because when it is hypertonic and when it is hypotonic, there is a possibility of cell death due to bursting because of the hypotonic environment inside the cell, and due to shrinking because of the hypertonic environment outside the cell. So the concentration of glucose must be maintained because of that. So when cells burst, it implies that there is a possibility of you dying. When the cells shrink, still there is a possibility of the organism dying. For example, I want to give you a task. If you plant two bean seeds and germinate into seedlings, giving them the same conditions in a container equal to those two containers, container A and container B. So after the growth of seedlings, maybe after like 2 weeks, you start watering them. One container A, start watering it with salty water, and then container B, just put too much water, or just put a little water, and then start observing them after like 3 days. You find out that after 3 days, the container which you have been watering salty water, we are going to see the leaves or the whole plant starting to die. Why is it dying? The cells are shrinking. It is going to dry out. It implies that it lost water to the surrounding environment because you created an hypertonic environment around it. Now the other plant is going to continue growing because the environment is not so hypertonic, or it is actually, it may even be hypotonic, but there's a possibility of it dying because if there is too much water again, it implies that the environment is going to be more hypotonic because you have diluted the existing salts which would be surrounding the plant. But of course, when you give it little water, it will cause it to continue growing, implying that too much of anything becomes bad. So too much solids become bad, and very little solids also become bad. Very little solids implies too much water. So we'll go to body temperature. The body temperature is maintained at a certain range between 36.5 to around 37.5. So beyond that range, you'll find out that the body activities are going to be distorted, or we can find out that the body activities are going to be disorganized. For example, which body activities may be disorganized? If the body temperature is very below 37 degrees Celsius, for example, if it goes to 20 degrees Celsius and below, or even 37 and below. So as the temperature starts falling down, you find out that the enzyme activity is also going to stop, or it is going to reduce. Why is it reducing? Because enzymes at a very low temperature, they become totally inactive. So when they become inactive, it implies that the body functioning may not functioning of the body may go down, because enzymes break down food, of which that food is one which is needed to form energy. Besides that, for the case of proteins, once they're broken down, they are body building foods and etc. and etc. So if you leave alone average temperature, of course, if it goes beyond and it goes like around 38, 39, 40 and beyond, we find out that enzyme denaturing is also going to take place. Enzymes will become totally denatured, and we may find out that chemical digestion of food may not easily take place. So as we go to number, that's why if you have to look at available body temperature, it is not advisable for you to take cold drinks when you are eating food. So though very many people are used to taking them as you are eating, because the coldness is going to lower the temperature which is in the stomach, and then the enzymes which are found in the stomach, for example, pepsin and renin, may not easily work very well because of the low temperatures. So it is very advisable to take the drinks later on after eating than you taking, for example, the cold water as you are eating. So we'll go to factor number 3 that should be kept constant. We have what we call pH. pH, you have 3 pH levels. That is the acidity, alkalinity, and then the neutral pH. So if you look at 7.4, the blood pH should be kept around 7.4, around 7 to 7.4. It can even be 6.9, 6.9 to around 7.4. And now 7.4 is the best pH for the blood. So if you find out it's very close to neutral, the neutral pH, so it means that if the pH goes to acidic or goes to alkaline, what happens? The body cells may be destroyed, or it can even denature the enzymes which are found in our bodies. So it implies that the blood pH should be kept at around a neutral pH. So it implies that when you get an organism, for example, you dip it in an acidic pH, you find out that that organism may possibly die. Though our enzymes, some of them, they may work at different pH levels, but now the blood pH affects the cells, for example, the white blood cells, the red blood cells, and etc. So the blood pH should be maintained at a pH at a neutral level. Why? It may affect the functioning of the body cells. But now for the case of the enzymes, some enzymes work best at neutral pH, others work, for example, at neutral pH you can find amylase, and then acidic pH we may find pepsin found in the stomach, because in the stomach produces alcoholic acid. Then when you go to the duodenum will produce bile that is alkaline. So it implies that the enzymes which are found in the duodenum, for example, trypsin, we have pancreatic amylase, pancreatic lipase, all those enzymes are going to require an alkaline pH. But now the pH in the blood must be kept constant at around that level, so that does not affect the normal functioning of the cells. Point number 4, we have what we call blood pressure. Blood pressure should also be kept constant. For example, at around 120, for the case of the high blood systolic value, and then the systolic value around 80 millimeters mercury Hg. So if the blood pressure goes beyond normal, again, it may affect the normal functioning of the body. Of course, we are going to see factor by factor, looking at how they can be maintained within the body. So the blood pressure, that is the speed at which blood moves within our blood vessels. So that one can also be determined by the normal pumping, by the pumping of the heart. The heart, normally for a normal person, an adult person, it always pumps around 72 beats a minute. So, but you may find it may go below, or it may go so high. Then it means that when it goes so below, then the blood pressure goes low. When it goes so high, then the blood pressure becomes so high. It implies that when it goes beyond the normal range, we find out that the body is going to be affected, and it can even cause death. So what point number 5? So we have 1, 2, 3, 4, 5. Concentration of carbon dioxide and other waste products of metabolism. So carbon dioxide itself, when it combines with the water inside the blood, it forms what we call carbonic acid. So carbonic acid increases, actually it lowers the pH of the blood. Actually, if it lowers the pH of the blood, it is going to affect the blood pH. So it implies that this carbon dioxide level must be maintained at a certain level, whereby it must not go so high and it must not go so below. So it implies that there are mechanisms which you are going to see that are going to expel this carbon dioxide outside in case we have too much of it. And then we can also find other waste products of metabolism. We shall see them. For example, urea, and etc. We are going to see how this can be maintained. Then we can see nitrogenous waste products from protein metabolism. Again, this one is so much linked from this because all of them are concerning metabolism. We shall see them together. Oh, I skipped osmotic pressure. Osmotic pressure, if we look at osmotic pressure, those are salts now found within the body. For example, maintenance of sodium ions, potassium ions, among others. So how do we maintain osmotic pressure within the body? So we shall go one by one, so that we can see how this can be maintained, so that we don't go beyond the normal. Another point, we have nitrogenous waste products. Okay, I talked about this. We have toxic substances liberated by pathogenic microorganisms. We have a lot of pathogenic microorganisms. For example, bacteria, dangerous bacteria, which is found in our bodies, they produce toxic substances. So those toxic substances, how can they be managed so that they can be liberated out of the body? So we look at the significance of homeostasis. We have looked at which factors that must be maintained constant within our body. So these factors, we are going to look at factor by factor, so that we can examine how are these maintained within our body. So we can look at the significance of homeostasis, of what advantage is maintaining the internal environment constant. Our point number one, optimal conditions for enzyme activity. I've told you, enzyme activity requires optimal conditions. For example, different enzymes work at a certain pH range. For example, pH range, for the case of the pepsin found in the stomach, pepsin works best at acidic conditions. And then in the duodenum, we find the alkaline conditions. For example, trypsin, again, works upon proteins, but trypsin works best in alkaline conditions. And then when you go to the small intestines, you shall find a neutral medium. So optimal conditions for enzyme activity. For example, again, you can look at temperature. So if the temperature goes beyond the normal, for example, 37 degrees Celsius, it goes to 40, enzymes are natural. If it goes below, you may find enzymes are going to be inactivated. Maintenance of body cells. Maintenance of body cells, for example, if blood becomes so hypertonic that there are too much salts, or we have too much salts inside the blood, the body cells, you may find out that they are going to lose water to the blood by osmosis. What is going to be the end result? The end result, the blood cells is to shrink. So they will shrink. So the blood cells like that, for example. So it is going to shrink due to the loss of water. It is going to cause cell death at that stage. So it is going to cause cell death. So because of the hypertonic nature of the blood. So the hypertonic nature of the blood is going to make cells lose water by osmosis. So a cell is going to shrink. But now, if there is a hypertonic environment within the blood, and the inside the cell is hypertonic, or inside the cell have more salts, what implies that this cell is going to be losing here water because of the hypertonic nature of blood. So blood has more salts than, so water is lost. So blood has more salts than the cell. But now if blood has less salts than the cell, water is going to come in. H2O. Water is going to come in, and what is going to be the end result? The end result is the cell is going to become so large, and at the end it is going to burst. It is going to burst. So once the cells burst and shrink, the cells are going to die at that stage. So the cells die when the conditions become abnormal. So maintenance of the body cells. Body cells are maintained by having, for example, the glucose levels maintained, because now that is the salt concentration. So we can go to point number 3, wider geographical range. So when the body systems are maintained constant, it means that an organism can easily move to various regions. For example, if I'm to go to London, for example, London during winter experiences very low temperatures, even to negative values. So those low temperatures, and I'm used to Africa, whereby the temperatures are a bit high. So the body is going to find ways of maintaining the body temperature, though you are living in cold conditions. So it implies that you can stay in wider geographical areas, or you can move to various places, and you can easily adapt. But if the body cannot easily maintain those conditions constant, it implies that you can hardly move to various geographical ranges, because different geographical ranges have different conditions, of which those conditions are strange. So if those conditions are strange, it implies that there are mechanisms that can be applied by the body so that they can be, you can easily survive within those conditions. So point number 3, regulates the body temperature. If the body temperature is regulated, we are going to see which mechanisms are provided so that the body temperature can be regulated. So if the body temperature is regulated, it means that the optimum temperature is going to be provided for example, enzyme activity. So body temperature is maintained at 37 degrees Celsius. However much you are in cold conditions, it will not go so below that range. That's why we are giving a range, not a specific value. So a specific value like 37, this is a wrong value. It is always a range. For example, 36.5 to 37.5, depending on the conditions. Because if it is too cold, the body temperature must not go below 36.5. If it is too hot, it must not go above 37.5 degrees Celsius. So we are maintaining within a certain range. That is the best range through which the body can easily function. So we can go to another point. So we have so far 1, 2, 3, 4. So point number 4. Point number 5 maintains water and salt balance. So why should we maintain water and salt balance? That we can maintain the osmotic pressure. Actually, this is going to be now the osmotic pressure, because osmotic pressure, that is salt balance. Now water and salt, there must be proper mixing of water and salt, so that the osmotic level must be maintained at a certain level whereby the cells will never shrink, the cells will never burst. So it must be maintained at an isotonic level. For example, blood or water inside blood, it must be having the same concentration of salts like those of the cells, because if the concentration changes, it implies that we are expecting shrinking or bursting of the cells. So we have maintaining pH. We look at maintaining blood glucose level. Blood glucose level, we are going to look at it in general. So blood glucose level must not exceed a certain value. It must not exceed 90 milligrams per 1 cubic centimeters. So we have maintaining pH. Of course, we looked at pH. pH can distort, or it can denature enzymes if at all it is beyond what is the required value. Prevents damage of tissues. Preventing damage of tissues, of course, we have talked about the cells being damaged by, for example, the salt concentration, etc. So we can look at the components of the homeostatic system. So the homeostatic system has 1, 2, 3, 4, 5, has mainly 5 components. What are those 5 components? The 5 components, number 1 is the reference point or the norm. The reference point or the norm, if I talk about a reference point, if at all I ask you a question and then you go and do your research. For example, you go to biological science to research. So biological science is going to be used as a reference point or a reference book. So that reference book, you are using it as a source of information. What does this book say about this? So the reference point, what is the norm, or what is required, what is the best condition for example, for that, for example, for blood glucose level, what is the best value for that, for blood glucose level within the body? So that is the reference. What is the normal blood pressure range? What is the normal pH range of, for example, blood? So this is the reference, the point, the reference point or the norm. So the normal condition. What should be the normal condition of blood pressure? What should be the normal condition of the blood glucose level? Number 2, we have what called the receptor stroke the detector. The receptor or the detector, it is the one which is going to detect a change. For example, in the skin, we have what called receptor cells. The receptor is going to detect a change. For example, if it is too hot, the skin is going to detect that the temperature is so high. Now, after detecting the receptor, within an office, you can find a receptionist. The receptionist is the one who is going to give, for example, he's going to be receiving visitors. And after receiving visitors, he's going to report to the boss that I have received so and so. So a receptionist, you have a receptor or the detector. For example, the eyes, those are receptors. Okay? They are receptors. Though they can sensory organs, but they are sensory organs which have receptors inside them. So they receive information. The receptor receives information. After receiving information, they're going to take it. Is this information good or bad? Does it require maintaining it or not? So we have what is called the control center or the controller. So the receptor is going to send information to the control center. So from the reference point, that is what is needed, what is the best value for example, blood glucose level. So after looking at that, the receptor is now going to get the information that this environment is so hot. For example, the skin is going to detect. Now this skin has receptor cells which are going to report through the sensory neurons to the control center or the controller. The controller mostly is going to be found in the what? In the brain or in the central nervous system. So at one point we shall also see the spinal cord getting involved in the reflex action. So from the control center after analyzing the information from the receptor, this information it has gone beyond what is required. Then the control center is going to send information to the effector. For example, the effector, those are going to be, for example, like the muscles. If you have the muscles and you have touched a hot object, the muscles may contract so that you can withdraw the hand. Or if the blood glucose level is going beyond get beyond the normal, the control center is going to detect, is going to send chemical signals to the effector. For example, the effectors can be the cells of Langerhans. For example, they are going to produce too much insulin. That insulin lowers the blood glucose level. We are going to see the blood glucose level. And then lastly, we are going to see the feedback loop. The feedback loop, that is the mechanism. That we are going to see the two types of mechanism. That is the negative feedback and the positive feedback. Now, the negative feedback or the negative feedback loop is going to be reversing, and then the positive feedback loop is going to be just increasing. As here, the word negative is going backwards, and then positive, it is increasing. For example, if the blood glucose levels go high, now we are going to have the negative feedback mechanism reducing the blood glucose levels. If the blood glucose levels goes so low, then we are going to see increasing. Now that is going to be positive feedback. So in general, we are having our homeostasis, maintaining the internal environment constant, looking at which factors must be maintained constant. Those are some of the factors. And then in general, if you look at the significance, those are some of the important points describing how important is homeostasis to the body, and then the components of the homeostatic system. Now in our next stage, we are going to look at the feedback mechanism. We're going to look at the main components of the homeostatic system, and then we shall begin with the feedback loop, or positive feedback and negative feedback. If you look at the feedback mechanisms are always employed just to make sure that the body remains in its normal state conditions. For example, if you look at the feedback mechanism, by definition, it is a control system in which the output of a process influences the process itself, helping the organism or the system regulate the internal conditions. For example, if you have a deviation in the blood glucose level, so we shall have an output that is going to bring the response back to normal so that the internal conditions can be maintained. So we have two types of feedback mechanisms that can resist or that can change deviations and put them back to normal. But the most common one is always the negative feedback, because the negative feedback, it is always employed at maintaining very many conditions which are going abnormal, or actually reversing them back to normal, because they're the most common conditions. So negative feedback occurs when a variable produces a response that reverses. As earlier I told you that negative feedback is all about reversing negative. You are going backwards. So why going backwards? Because there was a very big increase that required to be decreased so that we can put it at a certain specific value which is normal for the body functioning. So the response that reverses or reduces the original change, bringing it back to the normal. So that is negative feedback. So it is the most common type of feedback in the body, and it opposes the stimulus. Opposing the stimulus increases, but then negative feedback reduces. So it is always opposing the stimulus, and it is the most common type of feedback that we find in the body. So it is always the one which always maintains the body homeostasis. It is actually the one which is maintaining the internal environment constant. Why does it maintain the internal environment constant? Because if you look at the pH variation, normally it goes high. That is when it becomes too abnormal. So under rare circumstances that it will go low. So how does negative feedback occur? We have the stimulus. A stimulus bring a certain change. That stimulus is going to be received by the sensory organs. For example, the skin. For example, the skin is going to detect heat, thermoreceptors within the skin, and then the detectors are going to get the information. Now the receptors, the stimulus is, for example, heat or temperature, high temperature. So that the thermoreceptors are going to act as the detectors, which are going to detect that there is a deviation in the temperature. So the information is going to travel to the control center. The control center specifically can be the central nervous system or the brain. Sometimes you can look at the spinal cord when you are looking at the reflex actions. And then the control center is going to process information and then analyze and then send this to the effectors, that the effectors can reverse the deviation. The effectors can reverse the deviation. For example, you touch a hot object. When you remove your hand from the hot object, that is reversing. So the information that has been sent to the muscles of the hand is to contract to reverse. So that is how a negative feedback is going to occur. So examples of negative feedback, we have regulation of blood glucose levels, blood pressure, pH levels, regulation of temperature, among others. So you can look at the positive feedback mechanism. Positive feedback mechanism occurs when the change in the variable produces a response that reinforces or increases the original change. Positive, it is like now profit. Negative, it is like now a loss. So when you get a profit, you have added some income, you have added some money to the capital, to the input. So positive feedback mechanism, it is just increasing the original change. It is increasing it beyond the normal. So when does that happen? It is always less common. This kind of feedback mechanism is always less common. We can hardly find, but it is found we easily find it in our bodies, but it's not. It is very rare, like you can see in negative feedback mechanism. So it doesn't maintain homeostasis since it is just amplified. Since it is just amplified, it is just amplifying. It means that it is not as common as you can see in negative feedback. It continues until a specific point is reached. So it continues until a set goal is reached at that stage. So positive feedback, there is always a limit to which it is going to stop, but it will just continue. So there is amplification or increasing on its effect. So how does it happen? The stimulus causes an initial change. The response amplifies the change. So we see that positive feedback, we are having amplification only, not to the normal, but it's just increasing because as we are going to see in the examples, and then the process continues until completion. So as it increases, it will not increase forever. At a certain point, it's going to be reached, whereby if what is required has been attained, it is going to stop. So we shall not say that positive feedback mechanism is disadvantageous. It is advantageous. It is required so much in the body, though it is not so common. For example, if you look at childbirth, childbirth occurs at a certain stage whereby when a mother is giving birth, we find out that we need continuous within the labor, continuous contractions and relaxations of the cervical muscle so that the baby can be released. We are going to look at childbirth in details. So it always continues until the baby is outside. Then those contractions and relaxations are going to stop. Blood clotting. Blood clotting, we can see that a network of is going to be formed or to the wounded area. At a certain point, when the wound dries, so blood clotting, of course, is going to stop once blood stops, but it's going to continue until we see the blood is no longer being released. So sneezing, you're also going to see sneezing that you get some irritation within the nasal cavity, and when that irritation comes, we are going to see someone is going to sneeze, expel out, for example, the mucus, and once that expulsion occurs, the sneezing will stop. So we have lactation. Lactation, that is breastfeeding. Breastfeeding, it is when once the baby stimulates the nerves and the nipples of the mom, you find out that the information is going to be sent to the hypothalamus, and then later on, prolactin, which is responsible for production of milk, is going to be highly released, and then the baby is going to release a lot of milk. Even by just the mother, by just mere seeing the baby, milk is going to be produced within lactation will occur actually. So if you look at fruit ripening, what is responsible for fruit ripening? That is ethylene. We shall see once ripening stops, and then ethylene will no longer be produced. So we have action potential. We shall see action potential during coordination, and it is also a positive feedback mechanism. We have ovulation stage. Ovulation stage, that is when the egg is released on the 14th day in women. So once the egg is released, so the luteinizing hormone, which is responsible for ovulation, is going to be stopped from getting released. Remember, within the ovary, we have follicles, which are responsible for the formation of the egg. First, follicles produce estrogen, which goes to the brain, and into the brain we have the brain is going to be triggered, so that hypothalamus, so that we have luteinizing hormone getting released. The luteinizing hormone is the one which is going to be responsible for the ovulation to take place, and very many other examples of positive feedback mechanisms, of which once the set goal is reached, the mechanism is going to stop. Other examples, you can have release of pepsin and trypsin. Pepsin is released in the stomach, and then trypsin is released in the duodenum. Remember, pepsin, once it is in the stomach, it is in an inactive form called pepsinogen. But once food reaches into the stomach, of course, it is going to activate the stomach glands to produce the intestinal juice containing HCl. HCl, or hydrochloric acid, is going to activate the pepsin, which is in its inactive form is called pepsinogen, so that it can become pepsin, which is active. And in the duodenum, we have what we call trypsin. Trypsin, in an active form, it is called trypsinogen, and then it is activated by an enzyme called enterokinase or enteropeptidase. So what we can just say is that feedback mechanisms are only two types. That is negative feedback and then positive feedback. Negative feedback mechanisms are the most common mechanisms that we would always find in the body. For example, we talked about blood glucose levels, temperature regulation, blood pH, osmotic potential, among others. All those are controlled by negative feedback mechanisms or negative feedback loops. So if you look at positive feedback, it is always aimed at amplifying a certain event, so that that event, once it ends, once the set target is reached, that event is going to stop immediately, and then we shall see, for example, an advantage of that. For example, if a woman stops giving birth, the baby gets out, we shall see that what has been occurring, the changes which have been occurring so that the baby can get out, they immediately stop. So that is all about the feedback mechanisms. That is all about the feedback mechanisms. In our next lesson, we shall look at the negative feedback mechanisms, how blood glucose level is controlled, temperature regulation, among others. So we shall also look at widely the positive feedback mechanisms. For example, childbirth, how does childbirth occur, blood clotting, among others. So beginning at the first negative feedback mechanism we can have in our bodies, that is the blood glucose regulation. So when blood glucose levels change, what does the body do? So this is what we're going to look at. So which type of feedback mechanism does it apply? So we see that this is a negative feedback mechanism that is involved at maintaining the blood glucose level at a constant level. So we need to know what is involved, which hormones are involved, which body parts are involved. So we need the liver. We are going to see the importance of the liver. We need the pancreas. We need the hormones. Those hormones, they are only two, that is insulin and then glucagon. So regulation is essential to ensure constant supply of energy to the body cells, especially the brain. So it implies that when the blood glucose levels go down, it means that, remember, glucose is the source of energy. So if it is very low, so we need to see that it is getting maintained so that there is constant supply of energy, especially to the brain, because the brain, it is almost like the engine, the driving force of the body. It controls everything that takes place within the body. So what is the normal range? We said that maintaining to the normal, it must just be a range. So if someone has blood glucose levels ranging from 70 to 110, that person is very normal. If they are below 70, this person is abnormal. So it means that there is something that is lacking so that the blood glucose levels can be maintained. But if the body is having the insulin and glucagon, those hormones, of course, they will work hand in hand so that the blood glucose level is maintained at that level, which is 70 to 110 milligrams per 1,000 cubic centimeters of blood. So it implies that if you go beyond this, you are likely to be having diabetes at that stage. So diabetes may lead to high blood glucose levels coming in if you have a level which is beyond this. So if you're having like 80, 70, 90, 100, you are at the normal range up to a maximum of 110. So it normally occurs at a certain point. Blood glucose levels can even surge or can go beyond normal under some conditions you're going to see later on. So blood glucose levels, when will they always go high? Number one, after a meal. After a meal, the blood glucose levels will go high. But when they go high, but I've just given a single example of after a meal. But there are also other cases when the blood glucose levels can go high. For example, when adrenaline hormone is released into the blood, you find out that adrenaline is released when you are in fear, when you get shocked, when something shocks you, when you want to fight. Adrenaline is released into the blood. What happens? The response is going to be producing too much glucose so that you can get energy, so that you can maybe fight or fly away or run away. So too much glucose is brought in, is released into the blood so that there is that energy you are going to get, however weak you may be, and then you may find yourself getting a lot of energy. So, but in case you have just taken a meal and the meal it is of a carbohydrate, what will happen? So the beta cells are found in the pancreas. So the pancreas, the role of the pancreas is going to be producing the cells which are producing beta cells, and you're also going to see the alpha cells. So the beta cells of the pancreas, they detect the rays. So they will realize that there is a lot of glucose in the blood after you taking a meal. So what will happen? The pancreas is going to secrete insulin, and is going to cause the following changes. So the pancreas is going to secrete insulin and is going to cause the following changes. Number one, increased uptake of glucose by the cells. Number two, increase. So once what happens at this stage? Why is it that, because now the glucose is within the blood, once the cells take that glucose inside them, you find out that that glucose is going to be broken down, especially where respiration takes place, that is in the mitochondria. You're going to have increased conversion of glucose to energy. So increased uptake of glucose by the cells. So the cells are going to use that glucose, that it can be processed to form energy. So once the cell is taking more of that glucose, the glucose levels are going to go up to the normal. So number two, stimulates the conversion of glucose to glycogen in the liver and muscles. So glycogen and glucose. So once it is converted into glycogen, the glycogen is not going to remain as glucose. That process is going to be called glucogenesis. Glucogenesis is the formation of glucose. So glycogen is not going to be like a glucose, how it is going to cause. It is not going to be actually solute at that stage. So it is going to just be stored in the liver, and then it cannot be released into the blood. So it is just going to be stored, and it will not be a solid like you can see in the glucose to increase the hypertonic nature of blood. So to increase the solute concentration in the blood. So glycogen itself, glucose after being converted into glycogen, the solute concentration or the glucose levels will reduce, and that process you can call it glycogenesis. Another point you can see, it increases the conversion of excess glucose to fats. It increases the conversion of excess glucose to fats. Normally, people who have too much fats, it doesn't mean that they take a lot of fats. No. If you take too much of the glucose, or you can take too much of the carbohydrates, it can increase the amount of fats that you have in the body. So increasing the amount of fats you have in the body, it comes as a result of the increased conversion of excess glucose to fats. So if you have increased the conversion of glucose to fats, it implies that people who always eat supper very late and then they sleep, those people are going to develop a lot of fats. Fats have a disadvantage and an advantage. The disadvantage in most cases is that they can reduce the size of the blood vessels, or the lumen of the blood vessels. And once that lumen is reduced, that can reduce the pathway for the blood, and it therefore increases the blood pressure. So these fats can be formed from glucose. So if those fats are formed from glucose, it implies that someone can get diseases like high blood pressure because of the formation of fats. So the beta cells always do that role. Once those fats are formed, someone can develop a lot of fats within the body, not only because it's taking lipids or fats. So another point, it inhibits the breakdown of glycogen. Remember, glycogen, once it is broken down, it can form glucose. So glycogen is broken down to form glucose, or glycogenesis, glycogenolysis. Glycogen is converted to glucose. Gluconeogenesis. Is it the process? Yeah. So that process, when glycogen is broken down, stimulates the conversion of glucose to glycogen. So it will not be, it is gluconeogenesis, not glycogenesis. So gluconeogenesis, that is the formation of glucose. Glycogenesis, that is the formation of glycogen. So what will be the end result? All these actions that have been done, have been aiming at getting glucose back to what? Back to normal. So what about when glucose is very high? The opposite is going to occur. Whatever has been taking place here, it is going to be the opposite. So instead of the beta cells, you're going to have alpha cells of the pancreas that are detecting. So the detectors are the alpha cells that are going to take that the blood glucose level is very high. When the blood glucose level is very low. So I made an error. So this is, I can correct that. So when the blood glucose level is very low, so it is supposed to be very low here. So when the blood glucose level is very low, the alpha cells are going to detect that the blood glucose level is very low, and then we are going to have the following changes. So alpha cells of the pancreas detect a fall in the blood glucose and produce glucagon. So the hormone, here we had insulin, and the hormone, when the blood glucose level is very low, we have glucagon. So what is the role of that glucagon? Glucagon is going to stimulate the breakdown of glycogen to form glucose. So glycogen more so is a polysaccharide at that stage. Once it is broken down, it is going to form glucose. So stimulates the breakdown of glycogen to form glucose, of which that process, you can call it glycogenolysis, or you can call it gluconeogenesis, the formation of glucose from glycogen. So we're having stimulation of glucose from non-carbohydrates. For example, we can call it gluconeogenesis still. Gluconeogenesis, we are forming glucose from non-carbohydrates. Non-carbohydrates can also be fats, for example, and other energy sources. For example, like we can also have proteins still. Any other food source, once it is converted into glucose, that is gluconeogenesis. But of course, again, the formation of glucose from glycogen still is gluconeogenesis, or you can call it glycogenolysis, though it is more likely to be glycogenolysis because it involves the breakdown of glycogen. It involves the breakdown of glycogen to form glucose. So we have looked at so far the pancreas, the hormones. The pancreas produces the beta cells and the alpha cells. In the pancreas, within it, we have the isolates of Langerhans, which are responsible for the production of beta cells and alpha cells. So the hormones, we have looked at two hormones, that is the insulin when the blood glucose is very high, and then glucagon when the blood glucose level is very what? Is very low. So what is remaining is the liver. What is the role of the liver? So the liver can act as a glucose reservoir. So in case blood glucose level is very low, what happens? So the liver can act as a glucose reservoir. The storage of excess glucose can also exist in the liver. So store as excess glucose as glycogen. So the liver can do that role still. Excess glucose, glycogen can be stored there, and then when it is needed, when glucose is needed, so the glycogen can be converted back to glucose. So it is as glucose when the blood glucose level fails. So we can see that all in all, the liver is very important in storing glucose. So it means that the liver is very rich in glucose level, in glucose levels. So once it is needed, it is always produced like that. So what happens when someone has high blood glucose levels beyond what is normal and the insulin levels are very low? Because at this stage, someone can get sick of diabetes. Diabetes is a very dangerous disease. Diabetes, for example, diabetes mellitus. Diabetes mellitus can be caused by very high blood glucose levels. Very high blood glucose levels are managed by the production of insulin, the insulin hormone. So insulin hormone, once it is overwhelmed by high blood glucose levels, it is going to bring diabetes mellitus. Diabetes mellitus, it is due to high blood glucose levels. So diabetes mellitus is a dangerous disease which can add the hill, of which we are going to be requiring artificial insulin throughout your entire life. So it may result into very many changes like poor wound healing, blood pressure, and etc. Poor wound healing, it implies that when you get a wound, for example, on the arm, it is very hard for the wound to heal, and which it may develop into cancer, or it may become cancerous. And the only solution is to amputate. It is to amputate. In common English, you can say cutting off, but biologically, you can say amputation of the hand. You may find people who have been who have no legs. Someone whose leg has been removed. So why is that leg has been removed? In most cases, it may not be because of an accident. It is just because someone got diabetes and it caused the poor wound healing. Why does it cause poor wound healing? Poor wound healing, it is just because the blood itself is highly hypertonic, and it may cause a lot of cell death. So the cell death, it implies that the cells which are responsible for the blood clotting, they may fail in their activity, and what happens, we shall see that those cells have died, and blood clotting is now going to be very poor. So it is a very challenging disease. So in the next meeting, we are going to look at another negative feedback mechanism. But of course, I can give you a task of researching the types of diabetes. So I have type 1 and type 2, and you see the other cause of all the types of diabetes. For example, we have diabetes insipidus, we have diabetes mellitus. So the two types of diabetes, we have type 1 and type 2, and one of them is diabetes mellitus and diabetes insipidus. I want you to put in the comment section which one is type 1, which one is type 2, and give me the cause and the effect of diabetes insipidus, and what are the symptoms of diabetes insipidus. That can be research work. So we can look at the next negative feedback mechanism that can be controlled. water balance in the body it is also regulated by negative feedback mechanism. In that case, the water levels may go high or they may go low. So once those are detected, what happens at that stage? So we can find out the organisms which are involved. You can see the hypothalamus. Hypothalamus contains osmoreceptors. Osmoreceptors comes from the word osmosis or osmotic receptors. So what detects the osmotic potential, because now the osmotic potential is responsible for whether water is very high or water is very low. For example, in the cells, if in the cells we have a lot of salts, so we expect more water to enter into the cells. If we have less salts, we have and outside we have more salts, then it means that water is going to get lost from the cells to the surrounding environment. So the pituitary gland, and we are looking at the posterior pituitary gland. It releases ADH, that is the antidiuretic hormone. Antidiuretic hormone is responsible for water balance. Then you have the kidneys. Kidneys are just water absorption in the kidney nephrones. So what happens when water is very low? So when water is very low, we can call it dehydration. Someone is feeling thirsty. What will happen? So we look at what detects that. Detection is going to be done by the osmoreceptors from the hypothalamus. So I've talked about the organs which are involved, and the hypothalamus is among them. So once the hypothalamus detects from the osmoreceptors in the hypothalamus is going to send information. What the osmoreceptors are going to send information to the posterior pituitary gland, which is going to release antidiuretic hormone. Of course, we know hormone is travel through blood. So this hormone, which is ADH, is going to move through the blood and then to the kidney nephrons. So before we go to the effects of the ADH, I have put a note here, and that note is giving us there is a word called angiotensin. I'm bringing angiotensin. Why am I not bringing it earlier? Because its role is an indirect role. Angiotensin is just a hormone. So let us just look at it briefly. From the juxtaglomerular apparatus, these are the cells which are just below the glomerulus. If you look at the glomerulus, in excretion or in the kidney nephrons, those cells before the glomerulus produce renin. This renin is going to convert angiotensinogen to angiotensin 1, of which angiotensin 1 is going to be converted to angiotensin 2 by SCE. SCE, it is just there writing the description in full. So what is the role of the angiotensin? Angiotensin has an indirect role that it does, but we can see that it doesn't directly doesn't affect the kidney on water permeability, or it doesn't act directly on kidney water permeability. But what are its roles? It stimulates the release of ADH. That is number one. So this ADH, that is an indirect role. This ADH, which is released to go into the blood, it is the angiotensin 2 which helps in that role. So the angiotensin 2 is going to stimulate angiotensin 2 is going to stimulate the release of ADH. So stimulates aldosterone. Aldosterone increases the osmotic potential of the kidney tubules. So if the osmotic potential is very high, of course we expect water to get into the kidney tubules. Increases thirsty. Of course, that is a feeling which is going to give an indication that you must take water. So, but this was just by the way, so we can get back to the effects of ADH. Though this information is also important, but it is indirect, so we can also fix it there. But now we can see the effects of ADH, because ADH is the hormone which directly affects the water balance in the body. So it increases the permeability of the distal tubules and the collecting ducts. The permeability of what? Can have the permeability of salts. Salts can enter into the ducts. So when they enter into the ducts, of course, there is going to be an osmotic difference between the surrounding environment and then the collecting ducts. So water is going to get absorbed to the collecting ducts by osmosis. So at that stage, more water is going to be absorbed into the blood. So if we are to have more water getting to the blood, it means that the environment surrounding the collecting ducts should be more we should be having a lot of salts than the collecting ducts, so the blood will just water will just cross. So if this is our kidney tubule, so outside we should have, for example, more sodium ions, so that water inside the kidney tubules can cross into the surrounding area. Of course, that surrounding area now we are going to have water getting into the blood. So smaller volumes of concentrated urine are produced. At that stage, you produce concentrated urine. That urine, which is so dark, actually, which looks like yellow. It is so concentrated. Thirst sensation is stimulated, so you start feeling thirsty because of that ADH. So normally, when the water levels are very, very, very low, when the water levels are very low, it means you take a little water. It implies that all these changes are going to take place within our body. But the one which you can easily feel is the thirst sensation, and maybe the small volumes of concentrated urine. You urinate less when you have less water, but when you have more water, you urinate frequently. So I'm putting the question: when your water level is very high, what will happen at that stage? When your water level is very high, what will happen at that stage? Of course, whatever is here is going to be the opposite. Whatever is here is going to be the opposite. But of course, we know very well that the release of the antidiuretic hormone is not going to be there, because an antidiuretic hormone is going to just increase the permeability of the distal convoluted tubule and the collecting ducts. So what will happen? So whatever that is here is going to be the opposite of when the water level is very high. So it is your task now again. Can you find out, can you change this to the opposite, then you can get the solutions. Because at this stage, if we say increase the permeability of the DCT, so it means it is going to be decreased, because if this hormone is not released, then this is going to be decreased. More water is absorbed into the blood, so it is going to be less water. Smaller volumes of concentrated urine, so someone is going to urinate frequently, and the urine is going to be dilute. So that thirsty sensation is not going to happen, and you have less water returning to the blood. So you can find out when water level is very high, what happens, how is that maintained, so that water levels are brought back to normal. So I'm leaving you that task. So if you have any questions, you can put them in the comment section. Thank you very much.